Eddie is
Oh,
speaking in a couple of weeks.
Emilie, huh?
Oh, great. Hey, Neil.
Hi.
Hi, Neil.
Hi.
You remember Emilie and Neil?
Vaguely. It's been a long time.
Yeah,
and,
10 years.
and a different name.
All right,
Yes.
right.
Yeah.
Still me, though.
Yeah. Hi, Emilie.
Hi.
Okay, today's speaker is Emilie Purvine, formerly Hogan, who will speak about how to be useful and good for humanity using mathematics. Thank you.
Well, thank you for the introduction and for the invitation. I am going to get my screen share going, and I apologize. I think I'm going to look mostly at the big screen, but because my screen sharing is a little bit messed up, but I'll try to look over here for the most part. It was nice to see a bunch of names of people that I remember from Rutgers and from this seminar. It's nice to be back in the experimental math seminar. I wish I was in front of a board giving a talk to you all, but you'll have to suffer through my PowerPoint. And near the end, I'll get to the practice part of the talk. I'll start with the theory, and then I'll get into a demo near the end. So I'll start off by thanking my collaborators. I work with a lot of really talented folks here at PNNL, Pacific Northwest National Laboratory, and also a few interns and some folks that have moved on from PNNL. But it's a big group of people. This work is not at all my own only. So I wanted to acknowledge all of those folks. So just a brief outline. I'm going to start by motivating the hypernetwork science with somewhat more familiar graphs and network science, and then I'm going to go into the application, the extension to hypergraphs, also probably familiar, but I'm going to do it from a data science perspective. I'm still a mathematician, but my job title is a data scientist these days, so I think about everything from the perspective of data. And then I'll end with a brief demo of some software that we've been developing to play around with hypergraphs. So in a nod to the experimental math seminar, we'll show some experiments on real data. All right, so graphs. I expect most of the audience, if not all of the audience, is familiar with graphs, at least from a graph theory perspective. But like I said, I'm motivated from a data perspective. And so I think of graphs as a way to model data when it has two-way relationships. So when you want to ask certain questions of data that's modeled, that's in that type of format, things like how are the entities connected as a whole? Not how are they connected as pairs, but as a big picture, how are they connected? Or how do the entities cluster in different ways? Then a graph might be the right thing to use. And it's to model these kinds of pairwise interactions. Graphs, of course, are composed of vertices and edges, and the set of edges is a subset of all pairs of vertices. This is for a simple graph. A directed graph would have the, AB is different from BA. So when you have a graph that comes from data, oftentimes you want to know something about the structure of the system that you're modeling. And so you ask certain questions, and some of them are maybe more of a graph theory flavor, but a lot of them are what we call sort of network science. I think of network science as when you're working on data, and graph theory when you're working on sort of the hypothetical graph with extremal properties and colorings and things like that. So in one of these real data graphs, we might want to ask questions about the degree or the degree distribution. So for each node or vertex, how many neighbors does it have? And then as a collection, what is the distribution of those degrees across the whole graph? I'm showing a picture here of sort of like a power law distribution, although it has some interesting features that might be properties of the data or might be artifacts of how the data was collected. This is not the degree distribution for this graph, just as a disclaimer. Beyond just looking at the properties of a single node, which is the degree, we might want to look at bigger properties like walks, paths, and diameter of the graph. So a walk, of course, is a sequence of vertices connected by edges. A path is a walk in which vertices and edges are not repeated, and the diameter is the longest path in the graph. So I'm guessing that the longest path is somewhere between these two vertices here, perhaps. Once you have a notion of walks and paths, you can have a notion of connected components on whether two vertices, or sorry, whether a group of vertices is fully connected by a path, or whether you've got groups of components of your graph that you can't get from one to the other using a walk. This graph, of course, has one connected component. And then centrality is a really good way of measuring properties, or I guess, yeah, properties of vertices that may depend on the whole network rather than just where it is, it's on its own. So 5 images that or 6 images that I grabbed from Wikipedia to show you a few different notions of centrality. The top left is betweenness centrality. And here we're measuring for each vertex, how many shortest paths is it on. If it's on a lot of shortest paths, it's sort of a bottleneck. Those are the red colored nodes. And if it's not on a lot of shortest paths, then it's not a bottleneck, and it's one of the blue colored nodes. Closeness centrality is the upper right, where we're measuring how close on average a particular vertex is to all the other vertices. Eigenvector centrality is, as it sounds, an eigenvector. It's the eigenvector for the adjacency matrix that's all positive. Each vertex gets a score from that eigenvector. Degree centrality is actually just the degree. I don't know why they call it degree centrality, but they do. Harmonic centrality is a notion of closeness centrality that allows for multiple connected components. And then CATS centrality is related to, for a particular vertex, how many vertices are reachable from it on a path, and you kind of weight them by how far away they are. So if you have a lot of vertices that are really close versus you can reach a lot of vertices, but they're all kind of far away. All right, so beyond centrality, we can measure clustering coefficient. So centrality oftentimes is looking at a bigger picture. Clustering coefficient is looking just at the neighborhood of a vertex. How many triangles are there at that particular vertex? And then you can also ask about how many triangles are there scattered throughout the whole graph. These triangles might indicate some amount of, I don't know, clustering among the graph, if there's a lot of triangles versus if there's not a lot of triangles. So that's just a flavor of network science. There's plenty of other things that people consider: number of edges, density, so number of edges divided by total number of possible edges, average distance. You can look at random graph models. So if I have some real data, can I simulate a graph that kind of looks like it in distribution using a random model? Sometimes people like to ask about link prediction. If you have a dynamic graph, you know, maybe friendships or something, can we predict who the next pair of friends is going to be? And network science is really helpful in a lot of applications. So just noting a couple of applications here. The first that I've worked on is a power grid. So you can model the power grid where vertices are power consumers, distribution centers, substations, generators, things like that. And then power lines are represented as edges between those elements, sort of an abstract picture at the top. And then this is an actual graph rendering of the Polish power grid. It's a transmission network, so not distribution to every individual home, but just the big high voltage transmission network at different voltage levels. And what we found in this study is that the structure of a power grid graph is very different from the structure of other kind of real world graphs. It has a lot more longer loops, higher diameter than things like social networks. And so we had to come up with a new random graph model that would simulate graphs that had those properties. So that was a really fun project. Computer networks also are easily modeled as graphs, either the physical connections between computers, the possible connections between computers. Can I SSH from one computer to another? Do I have the right credentials? That's what's going on down here. And then up here is a model of actual network flow, so network traffic going from one IP to another. You know, as you're browsing the internet or whatever you're doing, you're sending packets of information between computers. And these kinds of networks get really big really fast. This is only 2 minutes of a relatively small system, and already it's got 1,000 vertices or so.
Emilie, you remember that AT&T used to have a very large room,
yes,
which displayed the traffic across the whole of the AT&T network, the whole of the United States,
yeah,
in real time. Yeah.
Wow, that's cool. So a third example is in protein interactions. Within a protein, it's composed of a bunch of amino acids, and those amino acids can interact in each other, and that's what causes protein folding. And so if you can model the interactions and the strengths of the interactions, you might be able to figure out what the structure of the protein is in a minimum energy configuration. They like to fold into those states that are minimal in terms of energy. And so by modeling the network or the system in that way, you might be able to discover that situation. But those are all pairwise, and the pairwise makes sense in all of those cases. But pairwise, modeling things as pairwise interactions isn't always enough to really model your data. So co-authorship data, where two people are connected by an edge, if they collaborate, is great, but that's not really how collaboration works, right? We have groups of collaborators. And so really what we want is a collaboration network where we group people by, you know, maybe papers, authors on papers or something like that. Friendship or following relationships in social data, again, people form pairwise friendships, that's certainly true, but really people form social groups, and so groups are really more the right model there. And another example is proteins. You know, of course, within a protein, these amino acids are interacting in pairwise, but they're also probably interacting in multi-way ways. And then groups of proteins come together to form a pathway, whereas if one of those proteins were missing, nothing would happen, but because all of them are there, something happens. So in this case, we want to go beyond graphs, and so motivating now hypergraphs. Hypergraphs, this is almost the same slide as I showed for graphs, with the change from two-way to multi-way. Again, we want to identify connectivity and clustering, where hyperedge now can be an arbitrary subset of vertices. And this differs from what we often see in kind of pure math, I'll say, or combinatorial exploration of hypergraphs, because oftentimes people treat hypergraphs as k-uniform, which means that every edge has the same size. But in real data, that's just not realistic for the most part. There's probably some cases where it is, but for the most part, it's not. So we need a general definition, or we use, of course, a general definition of hypergraphs to explore our data. So a hypergraph is going to be formally a set of vertices and a family of edges, and we use family because we don't want to say set of edges, because multi-edges are seen. You know, in a co-authorship or collaboration network, two papers might be written by the exact same group of authors. So we want to allow for those multi-edges. And we don't want to say multi-set because we want each edge to have kind of a unique name. So for that reason, we go with a family. A graph, of course, is a hypergraph where all edges have size 2. And this definition allows for the case of empty edges, multi-edges, as I mentioned, isolated vertices, so vertices that aren't in any edge, and also redundant vertices, which I'll talk about during the demo, I think, when I talk about something called collapsing. So that's just a call forward to that. So there are many different ways you can represent a hypergraph. There's this Euler diagram where we put all the vertices down and then we circle the groups of vertices that correspond to hyperedges. You can look at a bipartite visualization of the hypergraph where all the vertices are on the left and all the edges are on the right, and you connect it in the obvious way. Hypergraphs are represented by an incidence matrix where the vertices are on the rows and the edges are on the columns. So every edge contains the vertices for which there's a 1 in the column. Of course, graphs have an incidence matrix as well, but in a graph, you have a restriction that every column has to sum to exactly 2. Every edge has 2 elements. We can have a set notation for our hypergraph. And then lastly, this is going to be another call forward to some discussion about topological properties of hypergraphs. So simplicial diagram, thinking towards a simplicial complex. Okay, so hypergraphs are, I'm sure you understand, are different from graphs in some very specific ways. One of those ways is duality. So here's the graph, or the hypergraph that I showed on the prior slide with its incidence matrix. But there's no reason why one couldn't just transpose that incidence matrix and get another hypergraph. That's called the dual hypergraph. You can kind of see this from the bipartite graph. If you just switch the 2 roles, you get a different hypergraph, right? In a graph, every graph has a dual, yes, but the dual of a graph is almost always a hypergraph. And so we don't typically talk about the dual of a graph, whereas in a hypergraph, they're both hypergraphs. And so it doesn't always make sense to talk about one or the other, but kind of to think about a hypergraph and its dual as almost the same object. We can also approximate hypergraphs with graphs by just taking what's called the 2-section or skeleton of the graph or the hypergraph. So every pair of nodes that's in an hyperedge gets an edge. So we kind of squash things down into just this one-dimensional graph structure. And of course, the dual has a squashing down or a 2-section as well. What I won't get into this, but if you know the concept of a line graph, the 2-section of the dual is identical to the line graph of the original. All right, so let's get into a little bit of discussion of data and what kind of data generate hypergraphs easily. There's 3 examples that I'm giving here, and a lot of the cases tend to fall into one of these 3 examples. So first is attributes. So if you have a collection of entities and a collection of attributes, and each entity has some collection of those attributes for itself, that's a good setup for a hypergraph. So in this case, these attributes aren't really related to each other, but they could be. And the other thing to point out is that John, Sally, and Gertie being in the same hyperedge because they have brown eyes, it doesn't mean that they interact in any way. It means that they have some property that's common among them. So if you build a hypergraph based on this kind of attribute system, you're going to interpret the kind of hypernetwork science that I'll get to in a different way than you would interpret it in these other cases. Joint relationships is another way to get a hypergraph, and in this case, you do kind of have an interaction. So here I have some data from Active DNS project, which I'll talk about later, mapping domain names, so internet website addresses to IP addresses, and it turns out that each domain name can have multiple IPs, and each IP can have multiple domains, and you can structure that as a hypergraph. And then lastly, this is just just any old real valued matrix can be filtered to give you a hypergraph. So this is biological data, which is an example later, where in this case we could consider filtering by positive values and giving 1 to positive values and 0 to negative values, and then we now have incidence matrices for hypergraphs, and we can build a hypergraph. So these are the types of data, attributes, relationships, and kind of numerical matrices where you can get hypergraphs. So now that we kind of know where we get them from, let's talk about the hypernetwork science. and the combinatorial structure. So these are all similar to what I had on the network science slides, but beefed up for the hypergraph setting. So of course, degree. Every vertex is in some set of hyperedges, but we can talk about now the size of the hyperedges. So this B is in 3 hyperedges, but it's in 2 that are size 2 and 1 that's size 3. And maybe that's different than A, who's also in 3 hyperedges, but 1 is size 2, 1 size 3, and 1 size 4. So it's sort of they have the same degree, but you can think of it in different ways. Edge size is another hypergraph property. This is something that is kind of meaningless in a graph because all edges are size 2. But here, of course, we can have edges that are larger. And in order to talk about the next 3, I'm going to dive into them a little bit more and talk about what we mean by walks, paths, diameters, and so forth in the context of vertices versus the context of edges. So in a graph, a walk is a walk is a walk. If I give you a sequence of vertices, you could return to me a sequence of edges that correspond to that sequence of vertices, because every pair of vertices, as long as you're in a simple graph, belong to at most one edge. And even if you're not in a simple graph, the edges that you could belong to are going to be basically the same edge. Similarly, if you hand me a sequence of edges, I could give you back a unique sequence of vertices that correspond to that same walk. And so they're really one-to-one, vertex walks and edge walks. But in a hypergraph, this is not the case. Every pair of vertices can belong to many edges. Here, A and C belong to 2 distinct different edges, E1 and E4. And pairs of edges have many vertices in common. Here, E1 and E2 intersect at B and C. And so we can't get that unique one-to-one-ness between a walk on a vertices and walks on edges. So a walk between edges is going to be a sequence of successively intersecting edges, and a walk between vertices is going to be a sequence of successively adjacent vertices. And I'm going to focus on the edge
Emilie,
case.
But can you model it as a graph with many multiple edges, or it's not?
Yeah, in a way, you could take basically every pair of vertices. You could the skeleton that I showed on a couple slides ago, but you lose something, and I'll show you what we lose on the next slide. So if you do that, if you just say, okay, I'm going to connect all of the vertices if they're in at least one edge, and you talk about a walk on that, or if you connect pairs of edges by kind of a meta edge by saying that there's an intersection, you lose the connectivity of the strength of connectivity. So we've come up with a notion of an S walk. So this is a sequence of edges such that pairwise in sequence, they intersect at size at least S. So this means that walks and paths in hypergraph have a width component as well as a length component. So in a graph, here's a sequence of 4 vertices or 3 edges. We think about that as a hypergraph. We get this picture here. And edgewise, this is a walk of length 2, because I'm jumping from one edge, 1 jump, 2 jumps. And it has a width of 1, right? Because every pair of edges in a graph intersect at exactly 1 vertex. But in a hypergraph, these pairs of intersecting edges could be bigger or smaller. So here's a path of length 2, edge path of length 2 with S equals 1. So the strength of it is 1, fairly weak. And here is a case where the intersections are size 3 and size 4. So the width of this path is 3. And so if we were to model this as just a graph, we would lose this strength of interactions. Does that answer your question? I hope.
Good.
Okay. So just as we build up in graphs from walks to paths to distances to components, we can do the same in a hypergraph. So an S path is an S walk where edges are not repeated. In this case, I could think of my walk as going from 5 to 3 to 1, back to 5 to 10, and then to 12. But if I want to make a path, I could remove that kind of detour and just go from 5 to 10 to 12. This is a 1 walk or 1 path because all my intersections are size 1, but I can easily find a 2 path that's longer, but all of the intersections now from purple to red to teal to blue to orange are size 2. So it turns out that 5 and 12 are connected by a low width path, but also a high width path. Distance is then the shortest S path between 2 edges. And then we can find connected components as a collection of edges so that, again, any pair are connected via an S path now. So here is a 3 component. It's only 2 edges and they're connected by 4 vertices. A 2 component obviously grows things out, but still leaves off some of these edges that are connected in only by an intersection of size 1. One thing to point out here, which is kind of interesting, is that in a hypergraph, these components, they are a partition of edges, but they are not a partition of vertices. So for example, edge 13 is a 2 component all on its own. It has vertices k and I, but the vertex I is also in this big 2 component. And so we aren't splitting the hypergraph up into disjoint components using this S component notion, but they're kind of overlappy, which is hard to wrap my head around, or it was at the beginning when I was doing this. Okay, again, building past distances, we can get to centralities now on hypergraphs. Again, with this sort of S stratification. So question for S closeness centrality is which nodes or edges, because we want to be edge-based in a hypergraph, are close to everything. So in a graph, the definition of closeness centrality is the reciprocal of the average distance. So for a given vertex, we take the average distance of that vertex to all other vertices and then take the reciprocal of the average. The harmonic closeness centrality is the reciprocal of the harmonic mean. And this allows for disconnected components because 1 over infinity, we treat that as 0 if 2 vertices are in different components. Whereas here, if you have an infinity distance, that just your closeness is 0 no matter what, which is not exciting. So in a hypergraph, we can compute the same thing. I'm just gonna, I need to introduce one little subset before I can give you the translation of these centralities to hypergraphs. So the E sub S is going to be the set of all edges of size at least S. And why are we doing that? Well, if your edge is size less than S, then you're not going to be S connected to anything. And so we kind of don't care about you. So the closeness centrality for a hypergraph is basically exactly the same thing, but I'm going to put a D sub S, so the S distance between edges E and F, be the average of that for all F in my E sub S set. And similarly, harmonic closeness down here. So here's a picture of the hypergraph now colored by harmonic closeness centrality for S equals 2. So notice that the edges that were not part of that big 2 component that were all on their own have this closeness centrality of 0 because they just are components all on their own. Similarly, betweenness centrality can be computed or defined in a graph. The betweenness centrality for a vertex V is the sum of, for each pair of other vertices S and T, how many shortest paths between S and T is my vertex V on, divided by how many shortest paths total between S and T. We could do the same thing for hypergraphs. And now we see the betweenness centrality coloring for this example hypergraph. So one example that we've used this betweenness and closeness centrality in recently is in biological data. Sort of timely, we were doing this project before the pandemic hit. We were using SARS data. And when we realized this pandemic was going to be a thing, we kind of ramped up our project a bit, realizing it might have some helpful outcomes for the current pandemic. So what we had was data called transcriptomics data, which is measuring the expression of genes within a cell in a particular sample. So you treat a sample, or you give a sample a virus, and then you pull it at different time points, and you analyze what is the gene expression at each one of these time points. And you compare that to a control, so an uninfected version of that same cell line. And the value that you get for a particular gene at a particular sample is the log2 of the expression of that gene in the sample divided by the expression of that gene in the control. So that's what's going on here. So a positive value means that it's expressed more in the virus sample, and a negative means it's expressed less than the control in the virus sample. So we build a hypergraph from this, kind of like I told you earlier, by doing a filtering, but the filtering is a little bit more complicated than just positive and negative. We take z-scores column-wise, and then we filter on z-score greater than 2, and we have a p-value criteria. Not terribly important to understand all of that. It's really more important to understand that in this case, my vertices are conditions or samples, my edges are genes, and my gene edge encloses all of the condition vertices for which that gene is significantly expressed when you compare it to a control. So that's the hypergraph. And the goal was to find genes that are central in host-responsive viral infection. And our hypothesis was that the hypergraph, or the hypernetwork science measures, would be able to find genes that are important to host response better than network science would. And to do this, to test this hypothesis, we use something called gene score enrichment analysis. And so sort of a pictorial example of what that is. So if I have a ranked list, so let's say this is the top closeness centrality, the second highest closeness centrality, and so forth, and I have a target set of genes. For us, these were all immune response genes. We want to know, are these immune response genes kind of all at the top of my ranked list? If so, then my gene set enrichment analysis would be a high value. If they're all at the bottom of my list, it'll be a low or negative value. And if they're kind of uniformly distributed throughout my list, then it's about around 0 for my enrichment analysis. So this bar chart is showing the enrichment score for hypergraph ranked lists. There's 3 of them, and they were all the green ones. Graph ranked lists, the blue ones, and a sort of simple average fold change for each gene. And so what this is showing is that the hypergraph, our hypothesis was basically proved that the hypergraph ranked lists did better than the gene ranked lists to identify things that are previously known to be immune related. Okay, so that's the paper on arXiv that we've submitted elsewhere, and a news story that might be of interest if you thought that was cool and you want to read more. These are your 2 references. So one other example is, as I mentioned, computer network analysis. And so this is the DNS use case from Active DNS project, where we got a whole pile of data that correlates domain names to IP addresses. And in this case, our nodes are going to be IP addresses, and our hyperedges are going to be domains. And here's just a sample of an incidence matrix for this particular hypergraph here at the bottom. On the outset, we kind of thought that perhaps these would be one-to-one, that domains and IPs would be one-to-one. Turns out that's not the case. A lot of times they are, but there are things like domain aliasing, like if you go to Google but you mistype it, it's still going to go to Google. Like with 1G, it's still going to go to Google. So when you have domain aliasing, that is the case where you have maybe a single IP that goes to multiple domains. But you also have hosting services where maybe one IP goes to many domains based on what packets of information are sent. And let's see, so we were interested in questions like, can we find abnormal IPs? Can we just do some anomaly detection? And then also, could we do targeted exploration of the data? So just for one day, this data was pretty large. The active DNS puts stuff in the format called Avro, and there's 1,200 files per day. We parsed them into CSV, and we did some data cleaning, and came up with a big hypergraph of almost 11 million IPs and just about 130 million domains. So that's quite large. We had a multiprocessor system that we did some analysis. We did some component analysis. This is the degree distribution and the edge size distribution. And I'll show you just briefly a little bit about the components. We computed 1, 2, and 3 components, and just kind of did some exploration around the hypergraph to see what we saw. So the maximum diameter of all the 2 components was this guy here, and it's consistent with a particular type of behavior. We don't know that it's this type of behavior, but it's consistent with behavior used by botnets to hide malicious content delivery sites and to deliver malware. Whether that's actually true, we don't know, but the behavior looked kind of like it. So I mentioned earlier topology. Way in the beginning of the talk, I mentioned topology. So I wanted to just bring that in here before I get to the demo. So topology is the study, of course, of shape of data. You can identify things like holes. And typically, if you've seen topological data analysis, it's used to analyze point clouds like these up here to figure out kind of maybe what underlying system were these points drawn from. Here maybe a circle, here maybe a disk, and here some sort of line, curved line segment. Because these hypergraphs have some nice structure, we thought, oh, well, what about, can we do this for hypergraphs? They're not topological spaces, but could we make topological spaces from them? And of course, you can build a simplicial complex from a hypergraph by replacing each k-way hyperedge with a k simplex, k minus 1. I always get the dimension 1 off. So a 2-way edge gives you an edge, a 3-way edge gives you a filled-in triangle, a 4-way edge gives you a tetrahedra, and so forth. Or maybe we could build a point cloud from hypergraphs, basically preserve information about the structure of the hypergraph without adding or removing too much structure. And when you do that, you can identify topological properties. So you can identify one-dimensional holes in the hypergraph. By treating them as a simplicial complex, you can identify 2-dimensional holes. So this hollow tetrahedra here corresponds to a hollow tetrahedra in the hypergraph. You could identify one component, sorry, 0-dimensional holes, which correspond to s equals 1 components. And really, why do we want to do this? It's maybe to identify hypotheses or the need for more data. So if, for example, if this was a collaboration network, we might see that A, B, and C pairwise had papers together, but 3-way, they haven't had a paper together. Perhaps we should suggest that they should write a paper together. So we looked for these holes in the DNS data and did find some. We don't know what it means quite yet, but we found some holes. This is an illustration of a hollow tetrahedra here among these 4 IPs, the 98, the 160, the 9, and the 88. Here, it's somewhere in this hypergraph mess. The difficulty of hypergraph visualization is high.
Emilie, is that something
anyways,
surprising? If you take a random thing, won't you get artifacts like this? So
yeah, that's a good question. That's a really good question. I honestly don't know. We haven't done a lot of the kind of null hypothesis testing to see if that's expected. Part of that is because random hypergraphs can be more difficult even than random graph models. And so building a random hypergraph to kind of hypothesis test whether we expect to get holes or not is a non-trivial problem on its own. But good suggestion. Thank you. Okay, so I'm going to pivot over to the demo. Let's see. Hoping that you're still seeing my screen. Are you seeing a Jupyter notebook now? I think.
yeah.
Yes.
Okay, fantastic. So this is a tool called HyperNetX, or it's a Python package called HyperNetX. And I'm just going to show you a little bit in the remaining few minutes that I have of the kinds of things that you can do with this package. It's built here at PNNL. So it has a GitHub page and a documentation page. So if you are interested in it, pip install HyperNetX, and then just import HyperNetX. You need a few other things sometimes. So the hypergraph that I'm going to build is from the Les Mis, a subset of the Les Mis hypergraph. You build it really easily from a dictionary in Python. So each scene has a bunch of characters in it. Some scenes are 2 characters, some scenes are multiple characters. We have built-in, you might have guessed from my slides, we have a built-in visualization tool. It's somewhat of a random layout, so you sometimes have to run it a couple times to get something that looks halfway decent. But it's a flexible visualization tool, and it allows you, for small hypergraphs at least, to kind of see what's going on. I mentioned earlier that every hypergraph has a dual, and so we can visualize and compute the dual very easily by just using the dual function, and can kind of prove to you that in fact it is doing the dual. So this is the incidence dictionary, or kind of the same thing as the incidence matrix for the original hypergraph here, and then for the dual down here. So for example, the hyperedge 0 had fn and th in it, and if I look for where 0 occurs down in the dual, it's in the hyperedges that correspond to fn and th. So proof by example, the dual function works. And then you can visualize, of course, the dual. Let's see if I can get a better one. Yeah, that's sort of better. And you can see that there's these kind of duplicate, what appear to be duplicate edges in here, and they're going to correspond to vertices that play the same role in the original hypergraph. Other simple ways to play with hypergraphs in HNX are looking at the nodes. I did this on purpose. This is our custom data structure called an entity set. This is underlying everything in the hypergraph. But if you want to just see the list of nodes, you can cast it as a list. And so this is all the nodes in the hypergraph. Similarly, all of the edges in the hypergraph. Now, if your edges are usually edges are more interesting than just numbers. And then the shape will give you the number of vertices and the number of edges. As I mentioned earlier, the degree, you can just define the degree as simply the number of hyperedges that a vertex is contained within. So up here, fn is this guy, which is difficult to see in this layout. I'll try another one. Here now you can see that it's only in 2 vertices, 2 edges. One is size 2 and one is size 3. And so if I said s equals 2 in my, this is looking at how many edges of size 2 or greater is my vertex in. So it's in 2 of size 2 or greater, but it's only 1 of size 3 or greater. If you would like to know what edges the node is in, you can use this memberships property. So this tells you that fn is in the edges 0 and 2. And then you can use size to find out the size of an edge. So the edge 3 is size 4, and you can use the edges function to find out what exactly those edges were. So the s components and s distance can also be computed really easily. The components returns a generator. So I'm doing s equals 2 here on the edges. And so you can see that the 2 components of this hypergraph are mostly singleton edges, but one pair of edges 3 and 4, which are these 2 edges up here. One is a subset of the other. Similarly, you can compute the vertex components. So this is on the vertex level now. You can compute the distance between any pair of vertices or edges. So I didn't talk really about vertex distance, but it's edge distance in the dual. And you can see what happens if 2 vertices are not s connected. So default s being 1. If I put in s equals 2, see that vertices 4 and 6, or edges 4 and 6 are not 2 connected, so the distance is infinity. You can compute the diameters. Unfortunately, if the diameter is infinity, you get an error. But to get around that, you can use this function, which is plural, edge diameters, plural, and it will break the hypergraph into components and compute the diameter of each component, giving you the maximum, the list of diameters, and then the list of components. So either node diameters or edge diameters. And we can compute centrality. So we have a betweenness centrality, a closeness centrality. These are on the forward, the original hypergraphs, and then betweenness and closeness on the dual. If you know anything about Les Mis, you might see that the people that have high closeness centrality are Javert and Jean Valjean, which are, you know, two of the main characters in the story. This is just a few scenes, so it's not a good representation of the centrality. And I also mentioned this notion of redundant vertices. That is the best illustration is up here, where I said that BR, CC, and CN kind of play the same role. They're in all the same set of edges, and so they're redundant in a way. You can collapse them into one vertex. Same thing with JU and CH. And we have a function to collapse nodes. And then you can visualize and see that it did the right thing, that it collapsed the right thing, the right vertices. I will skip that in the interest of time and go down to the biological example. So this is the same biological data that I was showing you in the slides. This is the real data. It consisted of 279 conditions and almost 10,000 genes. The way that we create the hypergraph is by doing this z-score. So I'm going to compute the z-score on all of the columns. So this is now the z-score matrix, or the top of it at least, just so that it's easier to work with in the centrality in a moment. I'm going to just grab a subset of that, so the first 10% of the rows and the first 10% of the columns. And you can build a hypergraph directly from one of these data frames by giving it the data frame, whether you want the vertices to be the columns or the rows, and then your filtering criteria, so the greater than 2 criteria here. This hypergraph now has 27 vertices, which is the number of columns that I asked for. Actually, it's one less the number of columns that I asked for, and only 479 hyperedges instead of the 976. That's because some of the 976 genes had no conditions in which their z-score was greater than 2. A couple of functions here that I won't go into, but what we found in the paper is that rather than computing S centrality, we computed average S centrality from S equals 1 all the way to the top. And that did better. It took into account more structure of the hypergraph than just that single S. So these are functions just to compute that. And this takes just a moment to compute the betweenness and closeness centrality on that subsample of the biological hypergraph. And when that is done, it will visualize. Once it gets past 1, it goes through much faster. Or it did earlier. This is the problem with doing a live demo. So I will leave it to do its thing because I think it'll finish in very quickly and some pictures will pop up. And maybe I will stop there, although I will leave you at my kind of summary slide that we have. This is the links again for our Python package. And the Hypernetwork science is a great way to explore data in this kind of multi-dimensional way. 3 papers that might be interesting for you to check out. This Hypernetwork science via hypergraph walks goes through a lot of the theory that I talked about quickly today. And these 2 papers have some good examples. This last one is the biological networks paper that we just posted on arXiv. See if those pictures came up yet. Nope, weird. So I will leave it here. If you are interested in, if you have students that are interested in working at PNNL, we have summer intern postings available. And we are probably soon going to be posting a job for a postdoc in topological data analysis. If you know anybody, we'd be interested. So I will quit there and open up for questions. Hopefully I hit
so
the
much.
48-minute mark.
And before question, I have a quick question. How easy is it to generate examples to use your nice package? For example, if I use Messinet to get all the co-authors and make a database, can you handle very big hypergraphs?
Good question. So it sort of depends. So in the HyperNetX package, I haven't had much problem. It takes time to run. So to do the centrality computation on a big hypergraph takes a lot longer, as you might imagine, than on a small hypergraph. I would say on the order of 10,000 vertices and maybe 1,000 edges or 5,000 edges is doable on a small computer like a laptop if you're willing to wait. On a cluster, it'll go much faster. We also have other software that we're working on that is more scalable, and we have some backends that we're working on to plug C++ into HyperNetX that will do seamless and make things scale up a lot faster. So if you follow HyperNetX on GitHub, you might see those come out in the next 6 months or so.
Emilie, I have a question.
Yeah, go ahead.
I'm super impressed by the package's graph visualization techniques. Maybe you're familiar with NetworkX, the Python package, which has like atrocious visualization techniques. I kind of wonder, as like a technical point, how tricky was it to get these very pretty visualizations implemented?
Great question. And I need to give all that credit to Dustin Arendt here at PNNL. Dustin Arendt was the lead visualization person for the package, and Brenda Pregastis, which she might have joined. I don't know if she was able to make it. Doesn't look like it. Brenda Pregastis, also here at PNNL, is our lead Python developer she's a mathematician and lead developer. So those two worked tirelessly to get the visualizations to where they are. Dustin, here they are, the things that I was waiting for. So he used matplotlib. I think some NetworkX actually under the hood was used to do these visualizations. These are illustrations to show why it's so hard to do hypergraph visualization. But basically, I think he did a bipartite graph version of the hypergraph, and then removed the edge nodes and drew rubber bands around the collection of vertices for the hyperedge. So there are problems, like I don't actually know if all of these vertices are where they're supposed to be, but it works fairly well for at least small hypergraphs.
Are there any other questions?
Yes, I have a question. So, Emilie, are there scenarios in practice where it's desirable to have directed hyper edges?
Yes, thank you for asking that question. Yeah, there are a lot of scenarios. So biology is a good example where you have reactions, or chemistry, where you have reactions that have a group of inputs and a group of outputs to the reaction. And so that would definitely be a directed hyperedge. There are two kind of extreme types of directed hyperedges. One is what we think of like as a partition, a bipartition directed, where you have a group of inputs. You have a hyperedge, and half of some portion of them are noted as the ins, and the other portion of them are noted as the outs. That's one type of directed hypergraph. Another type of directed hypergraph is an ordered hypergraph. So for every edge, there is a sequence. So the vertices can be ordered 1, 2, 3, 4, 5, up to the size of the edge. And that's basically a full permutation of the vertices. Either of those, or something in between, like a poset directed hyperedge, could be thought of as an interpretation of a directed hypergraph, and they are definitely useful. We don't have the capability in HNX yet, but it's on our wish list.
Thank you.
I have a question.
Yep.
Is there a natural definition for the entropy of hypergraphs, and how does it relate to other measures of entropy?
Good question. And honestly, I don't know. So I've dealt with entropy of partitions, but a hypergraph is not a partition. So I'm not sure I would imagine there probably somebody somewhere has probably defined entropy of a hypergraph. And there's probably multiple ways of thinking about it, right? You have a degree distribution, so you could look at the entropy of the degree distribution. You could look at the entropy of the edge size distribution. So I guess the short answer is I don't know. The long answer is probably there's a lot of things you could compute the entropy of for a hypergraph. Thanks.
Any other questions? Well, let's thank Emilie again. Thank you.
Thank you.
Thank you. Bye. Great talk. Thank you.
Doron, are we going to the Indian?
Virtual Indian.
I wish.
One day, Emilie, we hope to get you in real life.
That would be great.
But thanks to you.
Wonderful talk. Thank you.
Pandemics, maybe you fight it.
We'll see. We're trying. Well, thanks for the invitation.
That's really amazing. So, well, what you do is relevant to the current pandemic.
Potentially, yeah. We were doing some work to try to identify genes that people might not have known previously as influential for other SARS outbreaks that might be important in this new SARS outbreak. I mean, that's a while before it actually makes an impact, because you have to identify the gene, and then you have to say, okay, what are the therapeutic targets that can regulate that gene? How can we make that into a drug? How can we test it? And so there's a lot of steps along the way, but that's a possibility.
So potentially you can help the pandemic.
Maybe someday.
That's good.
Yeah. All right. Thank you.
Well, thanks so much. This ends this meeting. Thank you.